So I told you about the reproduction, three types of reproduction in fungi. That is vegetative propagation. It is by the formation of uh, the fragmentation process, or it can be by binary fission or budding. That is the vegetative propagation of reproduction in fungi. Second method of reproduction in fungi is asexual reproduction, which involves formation of asexual spores like conidia, the sporangiospores, and the zoospores. They are produced in case of asexual reproduction. Sexual reproduction, there is a formation of the sexual spores, that is oospores, ascospores, and basidiospores. So in today's class, we are discussing about the uh, various steps involved in the sexual reproduction of fungi. The three steps which are involved in the sexual cycle of fungi is plasmogamy. Plasmogamy is the process of fusion of protoplasm of two motile or non-motile gametes. Okay, karyogamy is the fusion of two nuclei, and meiosis of zygote, which results in the formation of haploid spores. So these are the three steps involved in the sexual cycle of fungi. So if you look into this cycle, you can notice that the hyphae, they are going to produce these spores. So these spores, they are going to fuse, okay, or gametes, which might be motile or non-motile. They fuse at their protoplasm level. So the fusion of protoplasm, we call it as plasmogamy. And after this fusion of protoplasm, so there is a fusion of nuclei of this. So we call it as karyogamy. So plasmogamy is followed by karyogamy, that is the fusion of nucleus. Now they become diploid zygote. This diploid zygote immediately undergoes meiosis. So the diploid zygote undergoes meiosis to produce haploid spores. So these are the three steps that are involved in sexual cycle. So now in sexual reproduction, two haploid hyphae, they come together and fuse. In some fungi, fusion of two haploid cells immediately result in diploid cells. Okay, 2N. I just told you that these diagrams that are there, it is only for understanding. They're not there for your syllabus to practice them. Okay, it is only for understanding. So sexual reproduction in fungi involves two haploid hyphae coming together and fusing. So in some of this fungi, the fusion of two haploid cells immediately results in the fusion of diploid cells. So plasmogamy is immediately followed by the karyogamy. Okay. But in Ascomycetis and Basidiomycetus, the nucleus are not going to fuse. So they are found in a resting stage called as dikaryotic stage or dikaryiophase. That is the two nuclei per cell occurs. Okay, there is plasmogamy, but immediately they are not fusing. Instead, they form a dikaryophase where the two nuclei are present together per cell. So we call it as dikaryotic stage or dikaryophase. So such a condition, we call it as dikaryon. So the phase is called as dikaryophase or dikaryotic stage. So in case of ascomycetes and basidiomycetes, after the fusion of the protoplasm, the two nuclei don't immediately fuse. They stay in a resting stage. So wherein these two nuclei are present per cell. So we call this as dikaryotic stage or dikaryophase, and the condition is called as dikaryon. Later, this parental nuclei fuse and they are going to become diploid. So this is a delayed karyogamy in case of ascomycetis and basidiomycetes. So immediately after plasmogamy, the nuclei don't divide, rather they enter into a dikaryon phase, and then the nuclei, parent nuclei are going to fuse later and the cell becomes diploid. So fungi, they form fruiting bodies in which reduction division occurs. So the fruiting bodies in Ascomycetis, we call it as ascocarp. The fruiting body in Basidiomycetus, we call it as basidiocarp. Basidiocarp is the mushroom. So the edible part of mushroom is the basidiocarp. What we are eating is the fruiting body. Okay, so the reduction division occurs in the fruiting bodies, leading to the formation of haploid spores. In case of basidiocarp, they produce basidiospores, which are haploid. In case of ascocarp, they produce haploid ascospores. So now there is classification of fungi. The next part is classification of fungi. So based on the morphology of fungi, of mycelium and mode of spore formation and fruiting bodies. See three criterions they have taken for classification of fungi: morphology of mycelium, the mode of spore formation and fruiting bodies. The fungi are classified as Phycomyositis. So the bread mounds comes under the Phycomycetes. Ascomycetis, you have this ascocarp. Okay, the fruiting bodies of ascocarp you can notice, and yeast is also placed under Ascomycetis. Basidiomycetes, you have this mushroom, the button mushroom, oyster mushroom, milky mushrooms. They all come under Basidiomycetes. The fruiting body we call it as Basidiocarp. And Deuteromycetes includes those fungi in which sexual reproduction is not seen, or we have not studied it yet. Okay, or we have not yet discovered this process of sexual reproduction. Such fungi where the sexual reproduction is not seen or studied, we place them under Deuteromycetes. Deuteromycetes is also called as fungi imperfecti. See, the way based on the morphology of mycelium, the mode of spore formation, and fruiting bodies, the fungi are classified into four classes: Phycomycetes, which includes bread mold; Ascomycetes, which includes yeast, which includes a lot of other varieties; the Basidiomycetes, which includes mushrooms. So they are placed under the Basidiomycetes because they have the fruiting body basidiocarp and they produce basidiospores. The Deuteromycetes includes that group of fungi which does not have sexual reproduction, or the sexual reproduction process we have not yet studied. So we call them as Deuteromycetes, okay, or fungi imperfecti. So these are the four classes of fungi. So we'll study one by one each class. The Phycomycetes is called as lower fungi. Okay, the lower types of fungi. So generally, this Phycomycetes they occur in aquatic habitats and on decaying wood in moist and damp places, or as obligate parasites on plants. When you say obligate, it is compulsory parasite on plants. So the Phycomycetes are called as simple fungi. So they are found, majority of them are found in water, aquatic habitat, and on decaying wood in moist and damp places, or they are noticed as obligate parasites on plants. See, the mycelium is aseptate and they have cenocytic type of hyphae. See, the mycelium is aseptate, they don't have cross walls or septa, and their cenocytic hyphae is noticed in this Phycomycetes. Example for Phycomycetes is mucar. You can see this mucar here. Rhizopus, which is called as bread mold. Okay, you can notice it from this. Rhizopus, you take from the bread mold, you can study the rhizopus. Albugo candida, which causes the disease in this plant. So albugo candida, it causes the disease. So it's a parasitic fungi on mustard plant. Mustard is sasvi. So you should remember these examples because even in the board exams or CET or NEET, these examples will be given. So textbook quoted examples, scientific names you should remember. So it is necessary. See, Phycomycetes, they are called as lower fungi. Where are they found? Phycomycetes. They are found in aquatic habitat. They are found on decaying wood in moist and damp places, or this Phycomycetes are also found as obligate parasites, that is compulsory parasites on plants like mustard. It is having this Phycomycetes which infects the mustard plant is Albugo candida. So it's a parasitic fungi on mustard. And one more important aspect of Phycomyositis is the mycelium is aseptate and hyphae is cenocytic hyphae, you notice. Example for this fungi is mucar and rhizopus, which is found in bread, so the bread mold, and albugo candida, which causes the white patches on mustard leaves. So this it's a parasitic fungi on mustard, albugo. And Phycomycetes, they exhibit asexual reproduction as well as sexual reproduction. Again, these diagrams are only for understanding part of it. So you don't require to practice these diagrams. It is just to explain. Okay, so asexual reproduction is by, in case of Phycomycetes, they undergo asexual reproduction, which is by motile zoospores or non-motile aplanospores. So the non-motile we call it as aplanospores, the motile we call it as zoospores. So asexual reproduction in case of Phycomycetes is by the formation of motile zoospores or non-motile aplanospores. And these zoospores or aplanospores, they are produced in a cavity, and that cavity or sac-like structure we call it as sporangium. Okay, so the sexual reproduction in Phycomycetes, there is fusion of two gametes from zygospores. So there is fusion of two gametes to form zygote or zygospores. So the gametes that are seen in case of this Phycomycetes, they can be isogamous, that is both the gametes are similar in shape, structure and size, or they can be anisogamous, that is they are dissimilar in structure, size and shape, or oogamous, even they are dissimilar, but in oogamous you find that males are smaller gametes and they are motile, whereas the females produce lesser number of egg cell, they are large in size and they are non-motile. So this type we call it as oogamous type of gametes. Okay, so the gametes which are formed can be isogamous type, which are similar, or anisogamous type, where they are dissimilar, or oogamous type, where the female uh, egg cell will be large and non-motile. So that is the oogamous type. So that is about Phycomycetes. The second class that we have is Ascomycetis, which is commonly called as sac fungi. See, the examples of Ascomycetes are yeast, Saccharomyces, the generic name is Saccharomyces, then Penicillium notatum, from which the antibiotic penicillin was extracted. See, in Ascomycetes, you find unicellular fungi like yeast, Saccharomyces, or multicellular forms. You can also see like Penicillium notatum, it is multicellular fungi. The mycelium is branched and septate. See, the mycelium in case of this Ascomycetes or sac fungi, they have cross walls. So they are branched and septate. So the mycelium in case of the sac fungi or Ascomycetes, they are branched and septate. So they are saprophytic, decomposers, parasitic or coprophilous. See, the Ascomycetes, they are saprophytic, that is, they are found growing on dead and decaying organic remnants of plants or animals. They are decomposers, that is, they break down complex organic molecules into simple organic molecules, that is, they decay the remnants of the plants or animals. Then parasitic, that is pathogenic, they can cause diseases also. Okay, some of the sac fungi or coprophilous, that is, they are found growing on dung. So you find some of the mushrooms also growing on dung. So you cow dung, you might have noticed that you call it as Nycod, isn't it? So even the sac fungi, they are saprophytic or decomposers, or they are pathogenic, which causes disease, or they are coprophilous. Coprophilous implies, so the yeast fungi is not yeast, fungi is seen growing on dung. So the sac fungi are also coprophilous in nature, that is, they are found growing on dung. So the asexual reproduction. In case of Ascomycetis, it is by the formation of conidia. So conidia are produced exogenously on the special mycelium called conidiophores. So the conidia germinate to produce mycelium. So in case of sac fungi, the asexual reproduction is by formation of conidia by conidiophores, and the conidia germinates to produce mycelium. Okay. So whereas in case of the Phycomyositis, we found that they are producing motile zoospores or non-motile aplanospores, whereas in sac fungi, so the conidia are produced exogenously on a special mycelium called as conidiophores. Conidia germinates to produce the mycelium. So the sexual reproduction in case of Ascomycetis, it is by ascospores produced endogenously. See, the ascospores are produced endogenously means outside, within. It is produced within a sac-like structure called as asci. So singular is ascus. Okay, plural is asci. So the ascospores are produced endogenously, that is within the sac, like asci. Asci are arranged to form ascocarps, that is the fruiting body. See, this is the fruiting body that you can notice in the Ascomycetis. So they are going to produce ascospores, and these ascospores are produced endogenously in a sac-like structure called as asci. So many asci are arranged to form. You can see at the tip of it, asci are there. So they are labeled as asci, developing asci. So these asci are arranged to form ascocarps. So this fruiting body, we call it as ascocarp. That is the sexual reproduction. So the fruiting body is called as ascocarp. So the ascospores are produced within endogenously within the asci. So the examples for Ascomycetis, aspergillus, you can notice this aspergillus, claviceps, you can notice, which causes the smut disease in sorghum. Okay, claviceps purpurea, it causes a disease. Okay, sorghum. So then you find Neurospora. See, Neurospora is called as the Drosophila of plant kingdom. So this Neurospora, many genetic experiments are conducted on Neurospora. So that is the reason this Neurospora, which is a fungi placed under Ascomycetis, they are called as the uh, Drosophila of plant kingdom. So Neurospora are called as Drosophila of plant kingdom or Cinderella of plant genetics. So Drosophila is called as Cinderella of genetics. Okay, but Cinderella of plant genetics is Neurospora. So if you want to study about the plant genetics, so the most commonly used example is Neurospora. That is the reason they are also called as Drosophila of plant kingdom. So the edible part of this Ascomycetus are morels. They are all the edible part of Ascomycetus. Truffles are also the Ascomycetus, the fruiting body, that are all edible. Okay, so we don't consume this morels and truffles, but in foreign countries they have consumed this morels and truffles. Okay, European countries, American countries, they consume this morels and truffles. We just very few of us, even now recently, the habit of eating mushroom is becoming common, but earlier days there was no such habit of eating mushrooms. So the morels and truffles, which belongs to Ascomycetes, are edible, and they are delicacies eaten by the European people of European countries and the American countries. So they eat this morels and truffles, which are delicacies. So remember these examples of Ascomycetes. So aspergillus, then claviceps, which causes uh, the smut in the rye, that is uh, claviceps purpurea. Okay, then neurospora, they are going to be the Neurospora crassa is called as the Cinderella of plant genetics or Drosophila of plant genetics. Then the edible Ascomycetes are morels and truffles. The next class is Basidiomycetus. See, Basidiomycetus, So the edible mushrooms, uh, the oyster mushroom, the button mushroom, then milky mushrooms, they are all from this Basidiomycetes. So the fruiting body itself is what we call it as mushroom, and that is nothing but basidiocarp. Okay, so the examples of this Basidiomycetes are mushrooms. Okay, then you have this bracket fungi. On decaying wood, you might have noticed this sort of fungi growing on them, bracket fungi. Now is the monsoon season, and you can see this sort of puff fungi, puff balls, so which are all belonging to Basidiomycetes. You might find this puff balls growing, especially during this rainy season. If you visit this Gnanabharati campus, that is Bangalore University, at this season, you can notice a lot of puff balls are formed. Okay, so during the rainy season, you notice this puff balls which are formed. So these are examples of the Basidiomycetes. So the Basidiomycetes includes mushrooms, bracket fungi, and puff balls. So they grow in soil or on logs and trees, tree stumps, and in living plant bodies as parasites. Example, rusts and smuts. They are pathogenic in nature, so they cause disease in plants. Okay, the mycelium in case of the Basidiomycetes, similar to Ascomycetis, they are also branched and septate. So the mycelium in case of this Basidiomycetes is also branched and septate. See, Basidiomycetes, the asexual spores are generally not found, but vegetative reproduction by fragmentation is common in case of Basidiomycetes. So there is the asexual spores are not formed in the Basidiomycetes, but vegetative reproduction by fragmentation is noticed in this Basidiomycetes. Okay, fragmentation is breaking of hyphae, and each hyphae develops into a new uh, mycelium or new fungi. See, the sex organs in case of Basidiomycetes are absent, but plasmogamy occurs, that is fusion of two vegetative or protoplasm of two vegetative or somatic cells of different strains or genotypes. That fusion of protoplasm occurs, which we call it as plasmogamy. The resultant structure after plasmogamy, it is followed by dikaryophase, where it is in a resting stage, dikaryotic. Then later it fuses to form the zygote and they undergo meiosis. So the resultant structure where there is a fusion of two hyphae, vegetative or somatic cells of different strains or genotype by fusion of protoplasm, we call it as plasmogamy. So there is no fusion of nucleus yet. So they undergo a dikaryophase or dikaryotic phase. And this gives rise to the basidium. So after the dikaryophase, which is for a short duration, the karyogamy, that is the fusion of these two nuclei take place, and they form the zygote immediately. So immediately the zygote undergoes meiosis within the basidium. They undergo meiosis, producing within the basidium. This is happening. They produce four basidiospores exogenously, that is externally. Say ascospores were produced endogenously, that is within the asci. In basidium, they are produced exogenously, that is outside this basidium. So four basidiospores are produced exogenously. So the basidia are arranged in fruiting body called as the basidiocarp. See what you notice here, the gills in this region, you find the basidium and they have the basidiospores. So the stalk, we call it as stipe. Okay, so this is how when you dissect this gill region, you can notice this basidia. So the basidium will be producing exogenously, they are producing the basidiospores and they are released. The entire fruiting body, we call it as basidiocarp. So basidia are arranged in fruiting bodies called basidiocarps. Example is Agaricus bisporus, that is button mushroom. Ustilago, smut disease is caused by this Ustilago. Puccinia, so rust fungus. So they cause rust fungus in wheat. So this Puccinia, so they are all the pathogenic varieties of Basidiomycetus. Then the fourth class that I wanted to discuss for today was Deuteromycetes, which is also called as fungi imperfecti or imperfect fungi. So why they are commonly known as imperfecti fungi is because only their asexual or vegetative phases are known. So their sexual stages are not yet discovered, so that is the reason they are placed under this Deuteromycetes or imperfect fungi. If the scientists discover the sexual stages in the organism, they immediately place it either in Ascomycetis or Basidiomycetes. So in Deuteromycetes, it is also possible that asexual and vegetative stage have been given one name, placing under Deuteromycetes. So all the fungi which has asexual, only asexual and vegetative stage, they are all placed under Deuteromycetes. And the sexual stage fungi, they are all placed under another class, either Basidiomycetes or Ascomycetes. So when the linkages are established, the fungi are correctly identified and moved out of Deuteromycetes either to Ascomycetes or Basidiomycetes. So the Deuteromycetes, which is also called as imperfect fungi, it includes a group of fungi which has only vegetative and asexual reproductive stage. So they do not exhibit sexual stage or phase. So once if the scientists discover that the organisms which are placed in Deuteromycetes, they have sexual phase, then they can either place it by the type of sexual type, either they can place it in Ascomycetes or Basidiomycetes. So when the linkages are established, the fungi are correctly identified and they are moved out of Deuteromycetes either to Ascomycetes or Basidiomycetes. So all the fungi which do not where the sexual stage is not yet discovered or which do not exhibit sexual stage, they are placed under Deuteromycetes class, Deuteromycetes. So in Deuteromycetes, what do you notice? They reproduce only by asexual spores. There might be conidia. They have septate hypha. So the hyphae is septate, mycelium is septate and branched. See, some of these Deuteromycetes, they are saprophytic. They are found growing on dead and decaying organic remnants, or they might be pathogenic. So majority of the Deuteromycetes, they decompose litter, that is fallen leaves. So they are involved in decomposition of litter, and they also, this fungi imperfecti, they help in mineral cycling. So example for this Deuteromycetes are Alternaria, Coletotrichum, which causes the red rot in sugarcane, Trichoderma. So the Alternaria, Coletotrichum, Trichoderma are the examples of Deuteromycetes. There is also one Sarcospora arachidicola, which causes Tika disease in groundnut. They are also placed under Deuteromycetes. So the Sarcospora, which causes Tika disease of groundnut, wherein they have the spots, brown spots on the leaves of groundnut, and they reduce the yield of groundnut. So this particular the Tika disease of groundnut, it is caused by Sarcospora personata. There is also one more species, Sarcospora arachidicola. So if you study degree in botany, so you'll be coming across them. So they are also placed under this uh, Deuteromycetes. So these are the diagrams that they have given in your mucar, then the agaricus bisporus, aspergillus, okay, and the agaricus bisporus. So these are all the diagrams that they have given in your text. So that is sufficient for today's class. Okay, if you have any doubts or queries, please let me know. Do you have any doubt or queries?